Thank you very much for the kind introduction. My name is Tobias Berg. I'll present to you today some of our work on acute myeloid leukemia and some of the research plans that we have. In that context, obviously also introducing these research platforms that Kate has already mentioned. While the primary topic AML may not be in the core, let's put it that way, of your topics, these research platforms could be interesting, and there are a couple of collaborations already either started or in the process of being started, and that's why Kjetil invited me today to give a presentation a bit on the work that we're doing using these platforms. So I would like to start by introducing acute myeloid leukemia. It's actually the most common acute leukemia in the adult population. It arises either in a hematopoietic stem cell or multipotent progenitor cell that then gets transformed into a leukemic stem cell that has the same abilities to self-renew and also gives rise to numerous of these immature blast cells, which then outgrow the normal bone marrow cells and thereby lead to a deficiency of these cells, which is usually the clinical manifestation that the patients present with. It's unfortunately one of the deadliest diseases there are, and only about 21% of patients survive long term. And the main reason is that although we have some very effective treatments that work for younger patients, most patient population is actually above the age of 67. So the median age is 67 and can't tolerate these intensive treatments. And for these patients, we currently only have palliative treatments available. This is how one of these blast cells looks like under the microscope. And the acute myeloid leukemia as a term is actually defined by having more than 20% of them in the bone marrow. AML is, and that's something, is in a way a model disease. So a lot of the genetic work that has been done in cancer has originally been done in acute myeloid leukemia because it's kind of easy to get to living tumor cells that can then be investigated. And so a lot of that genetic work has shown certain genetic translocations, and AML has also been the first malignancy to be really fully sequenced. And so we now understand that AML is not just one disease, but actually many, many different ones. And that is due to mutations that occur in many, many different players. Some of them are signaling molecules, and many of them really play a role in epigenetic regulation. And this is something that we need later on. How does the treatment of AML work? The treatment of AML has been more or less unchanged for many, many years. So this treatment that I'll introduce to you has actually been around since the 70s of the last century. So when a patient with AML presents, we usually give an induction chemotherapy that consists of 3 days of daunorubicin, 7 days of cytarabine, which in our jargon we call 7+3. And this treatment is actually aimed at removing all of these blast cells and then allowing the outgrowth of normal bone marrow cells again. After that treatment, patients go through a phase of aplasia where they don't make white blood cells, they don't make red blood cells, they don't make platelets. And this is the difficult phase during which patients have to be inpatients and are treated on an inpatient basis. Once this is achieved, we call this a complete remission. But even though the complete remission is there, we can usually only see by conventional cytology methods when these blast cells are more than 5% in the marrow. So even though we get below 5% with this induction chemotherapy in the majority of patients, we still require additional treatments to eradicate residual disease. And this is mostly done with an intense consolidation regimen with high-dose cytarabine that is given over multiple cycles. In some patients, this can then eradicate even this residual disease and lead to a cure. Otherwise, if we don't do this consolidation treatment, almost universally all patients show an early relapse. And this is how the consolidation treatment looks like. It's given over multiple cycles. As this is not sufficient for most of the patients, we are also using allogeneic stem cell transplantation. And this is really a transplant from a different donor. So a donor receives GCSF to mobilize stem cells. These stem cells are then collected from the blood and are given to a patient who has previously been treated with a high-dose intensive chemotherapy. After this cell transplant, about 14 days later, these white blood cells, or the patient starts to make white blood cells and red blood cells and platelets again. And this is what we call engraftment. The interesting thing, or the trick about why the allogeneic stem cell transplant is so much more effective than just giving chemotherapy alone in eradicating the leukemia, is really that it's not just an intense chemotherapy treatment, but it's really also an immune treatment. Because with the stem cell transplant, we give a lot of T cells, some B cells as well, and NK cells, that can then recognize the leukemia cell in the recipient and eradicate residual leukemia cells, even though these may not they may not have been eradicated by the high-dose chemotherapy. Stem cell transplantation has actually been shown to be beneficial in all patients with intermediate and adverse subgroups of leukemias, and these patients are therefore standardly treated receive this treatment. So here again, when we classify the disease by different genetic groups, we see that we use chemoconsolidation for those patients who are in favorable risk groups, and an allogeneic stem cell transplant in intermediate and adverse risk groups. Our stem cell transplantation program is actually located at the Juravinski Cancer Center, and we are doing about 70 allogeneic stem cell transplants per year here. And the program has recently undergone an expansion to make this treatment available to even more patients, and that also led to a growth of our catchment area, which then probably will lead to an increase in the number of transplants that we are doing over the coming years. The biggest problem after transplant, and this is when you look at CIBMTR registry data, is the biggest problem is still, despite that treatment is very effective, many patients still relapse after a transplant. So there is actually the biggest problem, while there are some other problems as well, like infection or graft versus host disease, the biggest problem is relapse. And this is really the thing that with my research I'm trying to tackle or to approach. And one way of understanding, or one way of finding out who is prone to relapse, is to look at minimal residual disease or measurable residual disease before transplantation. Because there's data around that has shown that actually minimal residual disease after transplant can predict relapse, and patients who show minimal residual disease almost show the same rates of relapse like if they would have active disease at that time of their transplant. So we are collecting samples from AML patients at different time points of their treatment. So we collect initial samples, we collect samples after remission induction, and we collect samples after consolidation and also after the stem cell transplant. We are saving these samples in our cancer stem cell bank, and we're using them then for functional studies. So we are collecting these samples as live cells. And samples from this live cell bank, or at least these longitudinal samples, will then be analyzed both by flow cytometry as well as by quantitative PCR for the presence of minimal residual disease. And this will allow us to help establish a clinical workflow to measure minimal residual disease, but also do research on minimal residual disease using our single cell platforms that I'll introduce to you later on. And what can we do when we know that there is minimal residual disease there? We can use adapted transplant strategies, and this is an example from a transplant strategy that we used at the previous center where I worked at, where we used a intense conditioning regimen with fludarabine, melphalan, and TBI. And in this population cohort of MRD positive patients could show that even though their prognosis by conventional predictors would be actually fairly poor, we can be very effective with this treatment. There are also post-transplant maintenance strategies that can be applied that can further reduce the rate of relapse. And this is really what we could do already today in patients who are MRD positive. But we further want to understand what MRD is, or what's happening on the MRD level. And in that context, I would like to come back to the point that AML is really an epigenetic disease. And what do I mean by that? Because many of the mutations affected or many of the genes affected in AML are actually epigenetic regulators. So what is epigenetics? So every cell that we have in our body has about the same genetic makeup. And even though there is the same genetic makeup, there can be a very different outcome in the end. Similar to, like you see that here with the caterpillar and the butterfly. They both have the same genetic makeup, but they have very, very different phenotypes, obviously. And the same thing applies also to the cells in our bone marrow. So while these early immature cells have the same genetic makeup as here, this happy mature granulocyte, they have to go through different steps that involve the activity of transcription factors, but also epigenetic changes to undergo this change and this differentiation. So how epigenetics works is that the chromatin or the DNA is actually rolled on the histone molecules. And these histone molecules can be covalently modified, thereby influencing how tightly they are packed. And this then allows either transcription factors to go to that chromatin, or it doesn't allow the transcription factors to bind to that chromatin region when they are very tightly packed. And this then influences the activity of the gene and the activity of the expression of the gene. In our lab, we now have a technology, the 10x Chromium controller, by which we can look at individual cells and analyze actually their both epigenetic makeup using a technique called attack sequencing, but also their expression profile using RNA sequencing. And we are planning to use this platform to further understand what is happening on an epigenetic level when there are these residual cells, and how these residual cells, or how or which differences there are in these residual cells that made them more resistant to chemotherapy and that then make them give rise to relapse. And one of these factors that influences epigenetic composition is one that we can already target today. And this is a factor that we've done quite a bit of research in our lab on, and it's called LSD1, or lysine-specific demethylase 1. It's actually histone demethylase, so it catalyzes the demethylation of histone 3, lysine 4, and the demethylation of histone 3, lysine 9. And by affecting these two different histone modifications, they can actually have different outcomes. Whereas H3K4 dimethyl is found on active enhancers, so by removing this methylation, we actually keep this enhancer blocked. H3K9 dimethyl is actually found on repressed chromatin, so on heterochromatin. And actually LSD1 is an FAD-dependent monoamino oxidase. It's an enzyme, and it can be blocked by various inhibitors, and I'll come to that later on. And LSD1 has previously been shown to play a role in leukemic stem cells or in a certain subtype of leukemia, and to be able to make the cells more sensitive to a vitamin A derivative that is called alpha-transretinoic acid. So what we wanted to understand if this LSD1 plays a broader role in AML, and we were using two different murine model systems for that. One of them, both of them are actually based on the retroviral transduction of hematopoietic cells from the bone marrow using retroviruses that encode for two different oncogenes. One of them is called HOXA9 and MEIS1, and this is a combination of the two factors, and the other one is called MN1. So when the bone marrow cells are transduced with these factors, the cells can then grow in a cytokine-dependent fashion, so you can expand them in vitro, and when they are transplanted into mice, they then give rise to leukemia. But as these cells can be grown in a cytokine-dependent fashion, they give us plenty of material to work with. And so we've established this leukemia actually from a conditional LSD1 knockout mouse. So we could establish the leukemia in the presence of the LSD1, and then by treatment with 4-hydroxytamoxifen, we could knock out the LSD1 with quite a high efficiency. And we then looked at morphology and the differentiation by flow cytometry. And what we saw then was that after a certain period of time, it actually took 3 or 4 days until that effect started, the cells proliferated less and they showed a more differentiated phenotype. This was in particular due for one of the models that is called HOXA9-MEIS1 and not as much for the other model, MN1. While there was a similar effect on proliferation, the effect on differentiation was actually a lot less in this model. And when you then look at the differentiation pattern that arises, these cells actually start to express mature markers and they lose immature markers like c-KIT. But the marker profile that we have is actually quite interesting because it's characterized not by either granulocytic or monocytic marker, but by a combined expression of the two of them. So that these LSD1 or that the knockout of LSD1 actually induces an aberrant type of differentiation, but induces a differentiation that can be seen and that actually has an impact on the cells. We further wanted to understand what happens or which of these transcriptional effects that I previously mentioned of the LSD1 knockout actually plays the biggest role in that context. We therefore did RNA-seq to analyze the two models. And what we saw then upon the knockout of LSD1 is a massive increase in the expression of many, many different genes. And many of these are actually related to differentiation, whereas we saw very few genes only downregulated, which is an interesting thing, and that points towards a main role for really this H3K4 dimethylation in that context. We then further wanted to understand which transcription factors actually change their activity, so which transcription factors can now bind that then lead to this expression changes. So we used an in silico prediction algorithm to see which transcription factors there are, and it's mainly two of them that are changed in the strongest way. One of them is called GFI1 and the other one is called SPY1, which is also called PU1. While these two transcription factors are the same in both the HOXA9-MEIS1 as well as the MN1 model, there is another transcription factor, and that is called IRF8, that is actually different between the two models. We then looked at the differential expression of these factors, and when you then look at the expression levels here by quantitative PCR, you don't see a lot of change in the PU1 or the GFI1, but you see a strong change in GFI1b and IRF8, at least in the HOXA9-MEIS1 model. So why is it now that this GFI1 and the PU1 are still the strongest upregulated factors? And that is really due to the mechanism, because they mostly, or most of the peaks, or most of the binding sites for the GFI1 and for the PU1 are actually co-localized. And GFI1 has previously been shown, and here this is a co-IP again showing this, to bind to LSD1 or to recruit, or GFI1 can recruit LSD1, which led us to a model that essentially on these PU1 binding sites, there are co-localized binding sites with GFI1, where the GFI1 actually recruits the LSD1 to these sites and thereby inhibits differentiation and maintains self-renewal. Whereas when you knock out the LSD1, these sites again become active, and some of these sites are found, for example, in these genes GFI1 and GFI1b, and that then leads to differentiation. I now want to shift gears a little bit and talk about pharmacological inhibition of LSD1, and this can be done by different inhibitors. A lot of inhibitors actually have been synthesized for that molecule, but they mostly can be grouped in two different groups. One of them are FAD-targeted inhibitors, and these actually form a covalent binding to the coenzyme. And the first compound that this was shown for is actually a compound that has been used as an antidepressant a lot, and that's called tranylcypromine. And then there have been others that have been more recently synthesized, and these are all capable of doing the same thing. They form a covalent adduct to the FAD and thereby cause an irreversible inhibition of the LSD1. There are also non-covalent inhibitors that more block the enzymatic pocket, and these are mechanistically a little bit different. And so we compared these different inhibitors in their effect on these leukemic cells, and this is here now again in this HOXA9-MEIS1 model. And there you can see that when you treat with the covalent binding inhibitors, the irreversible inhibitors, so tranylcypromine and its derivatives, you can see this differentiation effect very similar to how it would look like, or how it looks like in the knockout. Whereas you don't see this effect even when you treat with high doses of these non-covalent inhibitors. So essentially what you need to do is you need to block the enzyme pocket in a different way than it's done by these non-covalent inhibitors. And actually, when we then also used our two identified target transcription factors, GFI1b and IRF8, it could also show that also these transcription factors are only modified really by these covalent binding inhibitors. So there is other data that shows that essentially these covalent binding inhibitors can interfere with the interaction of GFI1 and LSD1. So this is probably the reason why that is. And we wanted to understand further then if inhibiting the enzymatic activity of the LSD1 is really the mechanism we have to go after, or if it's more of these like binding effects of the LSD1, or more of these structural effects of the LSD1. And we therefore used a conditional knock-in of an enzyme that mutant, again generated the leukemia or our leukemia models in this model, and there we could induce again the knock-in of this enzyme that mutant by treating with 4-hydroxytamoxifen. When doing this, we still see some differentiation there. However, when we then put the cells into mice and compare the knockout with the conditional knock-in, we see that while the conditional knockout improves the survival of the mice, so when we're taking out the LSD1 completely, the conditional knock-in of that enzyme that mutant can't have that effect anymore. So in summary, the inhibition of LSD1 induces an aberrant differentiation in leukemias. It's leading to the reactivation of these activation of these PU1 and GFI1 binding sites, and this effect is mainly found with the irreversible inhibitors. Some of these targets in downstream are actually fairly interesting, in particular the IRF8, as this has been shown also to play an important role, for example, in dendritic cell differentiation. And some of the surface markers that we are seeing actually upon treatment with the LSD1 inhibitors actually resemble dendritic cells. So they start expressing, for example, co-stimulatory molecules. And this is why we are also very much interested in the immune effects of these LSD1 inhibitors. Another effect that we know that the LSD1 inhibitors can have is an effect on leukemic stem cells. And we have investigated that really using a limiting dilution assay. So we treated the cells in vitro with an LSD1 inhibitor and then actually transplanted decreasing numbers of cells into mice. And using Poisson statistics, this can then be used to estimate the frequency of leukemic stem cells. And when you then compare actually the treated cells with the untreated cells, you see that there is actually about a 2-log reduction in the content of leukemic stem cells. Recently, there's been a lot of data actually that these leukemic stem cells, or in this case, we measure them by the leukemia initiating activity, actually depend a lot on certain metabolic pathways. And in particular, they depend on oxidative phosphorylation. This has been shown in beautiful work from Craig Jordan's lab that has been published already in 2013, and then a follow-up work showing in 2018 that really oxidative phosphorylation is a very, very important process in these leukemic stem cells. So one of the factors or one of the treatments that can affect this oxidative phosphorylation is really a BCL2 inhibitor. So essentially by targeting mitochondria, it reduces this BCL2 inhibitor, it's called venetoclax, reduces the ability of the leukemic stem cells to undergo to do oxidative phosphorylation. And that then leads to a reduction in leukemic stem cell content. We therefore very much got interested in metabolic processes and wanted to characterize the metabolic response to LSD1 inhibition. And so I've teamed up still at the time in Frankfurt with the group in the lab of Harald Schwalbe, where they are doing MR spectroscopy for a lot of metabolites. So we measured a lot of different metabolites after induction of the LSD1 inhibition. And interestingly, could see a major decrease in the level of glucose in the cells, but also acetate and succinate, which is essentially one of or two metabolites downstream of the glucose. So this was pointing a bit towards an effect either on the Krebs cycle with this player here, succinate, but also could be an effect on glycolysis. And we therefore are studying these processes in detail. And what's very helpful to us in that context is that we actually have access to a seahorse metabolic analyzer. Because when the cell metabolizes glucose and does that by glycolysis, this then in the end leads to the generation of lactate, which leads to small pH changes, in a decrease in the pH. When the cell metabolizes the glucose through the Krebs cycle, it then in the end goes into oxidative phosphorylation, which requires oxygen. So this leads to a reduction in oxygen, or this leads to the cell using more oxygen. And the seahorse metabolic analyzer can really measure these two at the same time. So it can measure pH changes, but it can also measure O2 changes in close proximity to the cells. So, and this is one example that where we treated a cell line MON13 with venetoclax, and then could nicely confirm that we see a major reduction here in actually the oxidative phosphorylation. And something that I find really helpful, that I find really nice in that context, is to show actually these metabolic profiles with the oxygen consumption rate on the y-axis and the pH change, or the ECAR, on the x-axis. And there, in this case, for the venetoclax, you are seeing a reduction in the oxygen consumption rate, so reduction in oxidative phosphorylation, whereas there's hardly any reduction in glycolysis. And when you then look at the mitochondrial oxygen consumption rate and compare that to the glycolysis rate, you see that the glycolysis after treatment with the venetoclax plays a much more important role for the metabolism of the cell. Interestingly, we found a very different pattern when we treated with the LSD1 inhibitor. This mainly led to a reduction in glycolysis, so the ECAR went down, whereas the oxygen consumption rate of the cell stayed almost the same. So when you again look at the ratio of oxygen consumption rate and to glycolysis, you see that the dependency of the cell on the oxidative phosphorylation actually gets a lot higher. So that we then wanted to find out whether we can combine the two and then see a reduction in both of them at the same time. And first preliminary experiments show that this is the case. And there was also a very interesting effect when we, when Kanwal, the postdoc in the lab who is doing these experiments, set up this assay for the first time. And that was that when combining the two, he saw a major decrease in the viability of the cells. So by shutting down their metabolism completely, that seems to be one of the effects why this combination is quite effective. So in summary, LSD1 inhibition reduces glycolysis, and this points towards a synergism of LSD1 inhibition and BCL2 inhibition through a metabolic pathway. And with that, I would like to come to a bit of a conclusion about the work in my lab. So we're trying to understand the biology and monitor minimal residual disease. And we're investigating novel epigenetic treatments. And we want to do that in combination to actually improve outcomes for allogeneic stem cell transplant. And I've been very fortunate to find a few people who joined my lab and work on these very interesting projects. So Kanwal was working with me as a postdoc, but also 2 graduate students, Pradharni and Sarah, as well as 3 undergraduate students, Hugo, Vivian, and Ram, who are pursuing projects in this area. And none of that work would be possible without collaborators that I found here in the context of, in particular, establishing the cell bank, but also when now venturing into the single cell technology with Christine at the genomics facility, and also other people who then joined me here with my interest in these single cell sequencing studies. And I would like to mention again Ketil, but also Marc Larcher, who both expressed a strong interest in using this platform. And at last, I want to thank the funding bodies that funded this work, still during my time in Germany, but also here in Canada, in particular the Boris Family Foundation, but also the Canadian Foundation for Innovation, who gave us a grant to fund some of that infrastructure that we're using. And with that, I would like to conclude, and I'm happy to take questions.